Hello, everyone, and welcome. For the longest time, whenever we talked about the brain and how it learns and changes, we really only focused on one cell, the neuron, right? They were always the stars of the show. But what if I told you the brain is less of a solo act and much more of a dynamic duo? Today, we're going to dive deep into this crucial and honestly often overlooked partnership between our neurons and their absolutely essential partners, the glial cells.
So, let's start with a really cool piece of history. When scientists got a chance to examine slices of Albert Einstein's brain, they were hunting for the secret to his genius. Did he have more neurons? Maybe bigger neurons? The answer, though, was way more surprising, and it points us directly to what we're talking about today.
And here it is, the big reveal. The study found that in a specific part of his brain, an area that's absolutely critical for complex, abstract thought, Einstein's brain didn't have more neurons. What it did have was a significantly higher number of glial cells for every single neuron. This was one of the first major clues that these so-called support cells were doing a whole lot more than just gluing the brain together.
So, here's our game plan for today. To really get why glial cells are so important, we first have to build a solid foundation. We'll start with the classic story of synaptic plasticity. Then, we'll see how glia completely changed that story, which will lead us all the way to some really powerful implications for health and disease.
All right, let's jump in. At its very core, things like learning and memory happen because the connections between neurons, what we call synapses, can actually change how strong they are. This ability to change is what we call synaptic plasticity. It is the fundamental way the brain adapts, learns, and stores information.
Now, plasticity isn't just one thing. It happens on totally different timelines. On one side, you've got short-term plasticity. Think of this as a temporary little tweak in communication that lasts, a few minutes tops. It's all about adjusting the machinery that's already there. But for real lasting memories, we need long-term plasticity. This is a much bigger deal. It involves building entirely new machinery, making new proteins, changing genes, and sometimes even growing brand new synaptic connections. This is the real stuff of learning.
And the most famous example of this is something called long-term potentiation, or LTP. To put it simply, LTP is the process of making a synapse stronger. When two neurons fire together over and over, that connection between them gets stronger, faster, more efficient. This is the real science behind that classic saying, neurons that fire together, wire together.
Okay, now for your exams. You absolutely need to know this cascade. This is the molecular heart of LTP. It all comes down to a very special receptor, the NMDA receptor. Think of it like a bouncer at a club that has a very strict two-part rule. It only opens if 1. Glutamate is there, and 2. The neuron is already excited, depolarized. That excitement literally kicks out a magnesium ion that's blocking the door. And when that happens, calcium floods in. That rush of calcium is the trigger. It activates a bunch of enzymes, and here's the key part. Those enzymes push more AMPA receptors up to the cell surface. More receptors means a bigger reaction to the same signal. Boom, the connection is stronger. It's potentiated.
But here's the thing, you can't just keep strengthening synapses forever, right? The brain also has to have a way to weaken them, to turn the volume down. That's where long-term depression, or LTD, comes in, and it is just as important as LTP. It's what allows us to fine-tune our brain circuits and maybe even clear out old, useless memories to make space for new ones. It's the brain's use-it-or-lose-it principle in action.
Okay, so what we've just covered is the classic neuron-only story of plasticity. Now, let's bring in the rest of the cast. Because while neurons might be the actors on stage, we now know that glial cells are the directors, the lighting crew, the sound engineers. They are the entire production crew, and they are actively, powerfully shaping the whole performance.
Let's start with the cells that make myelin, the oligodendrocytes. We used to think of myelin as just static insulation, like the plastic coating on an electrical wire. But what if it's not static? What if it's dynamic? It turns out oligodendrocytes can actively change the thickness of that myelin wrapping based on how much a neuron is firing. This is a huge deal because changing the myelin changes how fast the signal travels down the axon. It's an incredible way to fine-tune the timing of entire brain circuits.
And this isn't just a theory. We can see it happening. Our experiences physically reshape our brain's wiring. Studies have shown that learning a new skill, like practicing the piano or even just juggling for a few weeks, causes measurable changes in the white matter of your brain, those myelinated superhighways. The very act of learning tells your brain to optimize its own wiring to get better at that new skill. How cool is that?
So we've seen how glia can change the brain's information highways. Now let's zoom right back into those individual intersections, the synapse itself. The old textbook model, right, was a simple two-way chat between one neuron and the next. But that model, well, it's missing someone. We now know there's a third, critical player in that conversation.
Today, we talk about the tripartite synapse. This model recognizes that third, essential member of the conversation, the astrocyte. The little arms of a single astrocyte can wrap around thousands of synapses, listening in on the neuronal chatter and, this is the critical part, talking back by releasing its own chemical signals. It is not a passive bystander. It is an active, influential member of the team.
So this new picture raises a massive question. If these glial cells are active participants at the synapse, just how much control do they have? Do they just sort of, influence the existing conversations, or can they actually decide which conversations get to happen in the first place?
And the answer is a huge, resounding yes. Glia are literally the sculptors of our brain circuits. Astrocytes release chemicals that are absolutely essential for new synapses to even form, but they also help prune away the connections we don't need. They can find weak or inactive synapses and basically put a delete me tag on them. Then another type of glial cell, the microglia, comes along, sees that tag, and physically gobbles up and removes the synapse. It's this beautiful, coordinated dance to constantly shape and refine the brain's wiring.
So, if this Neuron Glia partnership is so fundamental to how a healthy brain works, it stands to reason that when that partnership breaks down, things can go very, very wrong. And this is where all this basic science becomes incredibly relevant for you in understanding human disease.
And wow, the link to disease is just incredibly clear. Problems with myelin plasticity are being connected to everything from schizophrenia to depression. Dysfunctional astrocytes are a key feature of Alzheimer's, Parkinson's, and epilepsy. And when microglia, our synaptic pruners, go rogue, they can contribute to massive neurodegeneration. The common thread in all of this is a breakdown in glial function.
In a diseased brain, astrocytes can go through this traumatic transformation. They become what we call reactive. They get bigger, their gene expression, it's all messed up, and they can start pumping out inflammatory molecules that are actually toxic to the neurons they're supposed to be helping. They switch from being helpful partners to actively making the problem worse. So the crucial point, the big message I want you to take away from all this is this. We can no longer think about brain disorders as purely neuronal diseases. A breakdown in that complex, constant communication between neurons and their glial partners is emerging as a central problem in pretty much every major brain disease you can name.
And that leaves us with one final, really thought-provoking question. For decades, almost all of our attempts to treat brain disease have been focused on the neurons. But if glial cells are such key players in both keeping the brain healthy and in making it sick, could targeting them be the key to developing powerful new treatments? Could the next generation of drugs for Alzheimer's or depression be ones that fix this dynamic duo, that restore the healthy partnership in the brain? It's a really compelling thought, and it truly represents a major shift in how we're thinking about the future of neuroscience.